It's really a pleasure to be here. We recently moved closer to St. Louis. We're down in Southern Illinois, so use this opportunity. First opportunity I had to come up to Mobot and give a talk here. So really excited about that, and thanks for coming out today. I want to talk about fungal conservation, kind of where we are at the moment, some of the new data that we're using, opportunities for community scientists, MOMS members, whatever else, to participate in this, and then kind of end with a few stories of how we put the data to use. So without further ado, we're kind of having a fungal moment right now. It's pretty amazing. We've had, for the first time, a cover on Science magazine. IPBES is the international, what is it? Anyway, blanking out. Anyway, it is kind of the equivalent to the climate change, international climate change work. This was one that was done for sustainable use of wild species, and we got a fungus on the front cover, which is the first time they've actually included fungi on that. We've had public, kind of general information, New York Times, The Guardian, and then we get really crazy. So Vogue had this whole article on clothes that were influenced by fungal designs, which is pretty cool. And then, of course, we can't forget The Last of Us that just had zombie fungi taking over humans, destroying the world. So, fungi are really where it's at right now. And I think that's about time. And also, besides the kind of public thing, fungi are important, right? They form, they're good to eat. Others of them will kill you. They're important symbionts. They form mutualistic relationships with most plants, with many insect species, but they also are important plant pathogens and animal pathogens, including human pathogens. And they're being shown a major source of medicines, antibiotics, other medicines, and really now being shown to be critical for mitigating climate change. So super important. And in fact, life as we know it wouldn't exist without fungi. So there's also a lot of them. This is the best estimate currently. There's estimates up to 10 million species, but I buy this somewhere between 2.2 and 4 million species of fungi out there in the world. So here we got an important group of organisms. There's a lot of them. And yet we have, this was 1991 Science article talking about the disappearing mushrooms, another mass extinction. And most of you probably haven't heard about that. We haven't had the quite PR team that amphibians have had to really highlight it. It's probably not as desperate as with amphibians either, but we do know that fungi are under threat. And that shouldn't be a surprise because fungi face the same threats as animals and plants, whether it's habitat loss, loss of their symbiotic partner, pollution, unsustainable use, and more being impacted by climate change. So from a conservation perspective, really determining what species are thriving and which are in danger, they're rare, are they declining, it's really crucial to set priorities. We never have enough resources, we never have enough people, we never have enough money just to indiscriminately, randomly pick things. I want to do conservation on this. We really need to think of some way to prioritize this work. And for many funding institutions, many organizations, it is really the IUCN Red List that's used to prioritize this action. Unfortunately, the conservation status of the vast majority of fungi have yet to be assessed. And this really has greatly hindered including fungi in conservation action, access to funding through the conservation initiatives, policy, etc. So it's been a challenge, and so we've been really working to change that. So where are we? I said there's estimated 3 to 4 million species of fungi. Only 151 species of those approximately have been named. So we know maybe 4% of the estimated species of fungi on the Earth. And of that 151 species, we've only assessed 625 for their conservation status. So you can see we've got a lot of work to do. But we are making progress, because I said 625, but 10 years ago, we only had 3 species on the IUCN Red List, and really it's only been in the last 5 or 6 years that we've started to make that process. We've started to have enough capacity to do the work. And we are now kind of our accumulation curve is pretty steep right now. So we're happy where we are, but we got a lot of work to do. So before going on, just to make sure everybody's aware, what is a red list assessment? And it really is just a standardized process that works across all multicellular organisms, I should say, so plants, animals, and fungi, that evaluates the risk of extinction of a species. And they do this by looking at the change in population over time. With this idea is that if the population of a species is increasing or stable, it's probably not under threat, or at least not imminent threat. If it is declining, and the rapidity of this decline will help indicate how threatened this species might be. And so that's all this red list is talking about. So when we talk about red list, it's just an assessment, basically, of population change of a species over time. And so where are we? I said we're 625. Well, we did this paper last year. There was 597 when we did the paper. And when we looked at this, we just looked at the list, and there's different categories of threat, from least concern to vulnerable to endangered, critically endangered, extinct. We don't have any documented, at least on a red list, documented extinct species. We hear from people they think they're extinct, but they haven't made that critical study enough to actually say that's really extinct. But when we look at where we are in the species we have almost 50% of the species are in one of these threat categories. And those of you that have been involved with plants or animals know that 50% is like a crazy number of species that are under threat. It would move it towards one of the most endangered group of organisms on the planet. And we think that's due in large part, this high is the way we've been doing it. So to get started, we've been inviting knowledgeable colleagues to nominate species that they think are threatened. And so we're already starting off with the idea that we're going to have a higher percentage of threatened species than if we did this randomly. And we know there's biases in the sampling. So these are the species that have been done. The vast majority of them are mushrooms and puffballs, so big macro fungi that are soft. We do have a couple of, bracket tough things. But those are basidiomycetes, which is an important group of organisms. But the vast majority of fungi are ascomycetes. So that's where your yeasts are, and your molds, and your morels, and other things like that. And we've done very few ascomycetes so far, and most of the ascomycetes are lichenized fungi. So we really have a bias in what we've looked at so far. And not surprisingly, geographically, we have a major bias. With most of the species, this thing is simple. This is the number of species that have been assessed. That's the number that are threatened in one of the threatened categories. And you can see that most of it we know from North America, Eurasia, and South America, with almost nothing from sub-Saharan Africa, very little from Southeast Asia, and a smidgen from Australasia. So we got big geographic range. So when we look at these data, what do we have? We have bias in our sampling, only few that have been assessed, real bias in our geography. And so how are we going to address that? And why do we have those biases? And that's partially because to do a red list assessment, you need certain data, right? You need to know where the species is. It's really hard to say something's endangered if you don't even know where it occurs. So we need data on their distribution. I mentioned that the red list assessment really is looking at population trends. So you got to know some idea of what its population size is and some idea of what might be happening. You know, is it doing well or does it seem to be declining? And then there's a little bit of biology you need to know and ecology because that helps you understand why might be happening. And then we don't want this just to be a list of species that are in problem. We really want to provide information on what is the threat, right? And when possible, what is the solution, right? Because otherwise you just have a list of things that says, oh, woe is us, these species are in trouble, and we've made no statement on how we can try to mitigate those concerns. So those threats and solutions are really hard. And as you can guess, with a group of organisms that we only have 4% even named, we don't have data on all of this. So that's what we're working on, is how do we get more data? How do we accumulate these data so we can do these threat assessments? So what are some of the sources of information? We're sitting in this incredible building with, you know, one of the larger herbaria in the world. Well, for fungi, we have large fungal collections too. Not at the Missouri Botanic Garden, but we do have large collections in various parts of the world. And so those collections are incredibly important. They are our history of where specimens occurred. It's really a history of where collectors went and when, but they're super important. And we're getting more and more ability to mine the information in those collections because we have things like data portals. So for fungi, we have the mycological collection, the mycoportal, which now is a one-stop shop where you can search all of the fungal collections in North America, or almost all of them, know where they are, and get pictures of their label data, and all that kind of stuff. So all of a sudden, we now have access to this vast storage of critical historical data. And those are there's even bigger aggregators with the Global Biodiversity Infrastructure Facility, GBIF, and I can't read it right now, but there's around 38 million records in GBIF right now for fungi. But even though it's kind of a scattershot, and we have to look at it, and I don't believe all of these things in the middle of the ocean, but that's okay. But if you look at the red spots, you can see where do we know something, even from these records. Oh, we know something from North America, Eurasia, a little bit of Australia, and the rest of the world is either blank or yellow. So again, when we look at the accumulated data that are in our collection data, we still see we have big gaps. So because fungal diversity and distribution is so poorly known, there's really great opportunity for discovery. And really engaging community scientists, citizen scientists in the work is really one of our best options for, at scale, developing these information. Really understand where fungi are, where they're occurring, and if there are patterns that are showing how that's changing. This is not new. We've had important amateur mycologists forever, or at least for a long time. In North America, probably the most famous is Charles McElveen. Charles McElveen is a retired Army colonel, I believe, and in the 1880s, he was cruising around on his horse. He went past this place and he saw all these fungi out there, and he said, wow, wonder what these look like. They should be potentially edible. And so he looked at the data, and there was like 25 species that had been documented for North America that were edible. And so he took upon himself for the next 12 years of going around, mainly Pennsylvania, tasting fungi, eating fungi. He knew what he was doing. He did not die of mushroom poisoning, folks. He knew the deadly Amanitas. And he was very scientific about it. He'd eat something, wait a couple of days, and he'd only eat one species. He wouldn't mix things. He'd eat something, wait a couple of days to see what his reaction was. And, he suffered some, but he didn't die. And then in 1990, he published this monumental book, 1,000 North American Fungi. And in that, he both described the fungi and he described his experience eating those fungi. Okay? What was edible, what wasn't. Some of you that have been involved in publishing those, sometimes you try to get a little lazy. So most field guides since 1900 have basically cribbed off of his knowledge on what's edible and what's not. Trouble is, Charles McElveen had an iron stomach, and he could eat things that not everybody would want to do. But still, a great job. It was really monumental work, and we still benefit from that work of this retired Army officer. We have societies that are involved in fungi. So Boston Mycological Club, founded in 1895, says that they are the oldest continuous mushroom club in the world. There were some earlier ones in France and England, but they kind of had fits and stops and didn't do that. So they claim to be the oldest, which is great. The North American Mycological Association, which is our kind of field-oriented fungal association, was founded in 1967. And my guess is Maxine knows when MOMS was founded. I couldn't find that data. 87? Okay, so the Missouri Mycological Club organization was founded in 87. And then the Fungal Biodiversity Survey kind of started as the North American Fungal Flora in 2012 and then reinvented itself in 2020. So that's kind of our history of citizen science work in North America. And most of the time, that work has been pretty much self-driven by interest, right? And so clubs had maybe their species list or whatever else, and then along came iNaturalist and Mushroom Observer. All of a sudden, we had a way for people to post pictures and get identifications. But of course, while they were doing that, every time you take a picture on your phone, right, it's geo-referenced, right? So I can tell exactly where that picture was taken, right, because that data is with that. So all of a sudden, we have this incredible new data collection tool from iNaturalist that people are putting up. And just some numbers just always boggle my mind. There are about 9,500,000 records on iNaturalist of fungi. And the thing that boggles my mind the most is almost 700,000 observers. There's only 1,500 mycologists in the Mycological Society of America. And in NAMA, there's what, about 10,000 or so people, something like that. And here there's 700,000 people putting up pictures of mushrooms. How do we make use of that? How do we get that? Because, right, we know that it's the quality of the observation, amount of metadata, so all the other information that's with it, it varies greatly on those, right? Some of the pictures are great, some of them is, what is that? And some have information, and some, will tell you that, this was collected in a forest with oaks and whatever else, and some don't tell you anything. So the high quality observations, that's really the basis for our further work. So there's several groups, including the Fungal Diversity Survey, that's actually have tried to work on that. So the Fungal Diversity Database is actually now encouraging and facilitating high quality observations on the site. They tell you how to take pictures, they tell you what your data you need, try to update that. And there's actually a team of triagers that can go and provide quality control, check identifications. If it's a really cool fungus, they've actually go out and try to reach the person that posted it to try to get more information about that. So really trying to use this incredible resource of iNaturalist to do more. Now, those are kind of self-directed, kind of random, people go out, see something cool. But there's a couple of really interesting directed projects that are going on now. The first one was Fungal Map that was organized out of Royal Botanic Gardens, Victoria, in Melbourne, Australia, by Tom May. And what they did was, the first iteration was they just found 100 species, or named 100 species, gave some information, and sent field naturalists, amateur people out in the field to map what was going on, okay? Importantly, they also said where they didn't find it. because oftentimes we don't know, just because you don't have a record from that spot, did somebody look there or not? And just, this is probably not going to be shocking to anybody, but here's kind of the impact of citizen scientists, at least in Australia. This was their fungarium specimens, and you can see that, just like most herbaria, most species have only a couple of records, right? Because that wasn't the goal of the herbarium. The goal was to document diversity, not necessarily document frequency or anything like that. So most specimens in the fungarium in Victoria have, 1, 2, 4, maybe some teens. There's not much you can do to say about full distribution of a species with that. There's nothing you can say about change in population from that. Versus the fungi target map species, most of the species are 100, 1,000, or more. All of a sudden, when you have those kind of data, you can start talking about what's going on with that species. And so the impact of having targeted searches by interested amateurs, interested citizen scientists, really is amazing. And just, probably not the best example, but I like that Amanita, so I showed this one. The yellow dots are what was known, so there were 3 records of the species known for all of Australia, 2 from Western Australia, 1 from just this tip of Victoria. And then with new records, you can see it's really expanded into Tasmania, up the east coast, the southern coast, and really expanded the knowledge of what's going on with that. Another good project was the Lost and Found project that was organized by Royal Botanic Garden Kew. And what they did was they identified 100 species that had not been recorded in the UK for a long period of time and were presumed potentially to be extinct, or extirpated, I should say, from that. No longer in the UK. And they sent people out, they prepared some information for them. And what they did was they found 77 of these species. They also found a bunch of bycatch, other cool specimens that they had. But if they found 77, it meant that there was still, after 5 years of really directed collecting, all of these species were not recollected. So that, hey, there really is good information that those things are either extremely rare or no longer occur in the UK. And then there was these 4 that they called Lazarus species. They did find them for the first time after 50 years. They hadn't been seen in at least 50 years, so they rose from the dead. What was fascinating about this, of course, is the ones that they did find oftentimes were the most inconspicuous ones that nobody had bothered looking for, little microfungi, whatever else. Not surprising, no one had seen those. The ones that they didn't find were occasionally some really big, beautiful mushrooms that if, with that army of people out there, they would have found it, and they didn't recollect it. So we really have some confidence now that these species have been extirpated from the islands. And we're doing something similar here in the US. I'm not going to go in detail because there's other more interesting things to talk about. But Fundus has this West Coast Rare Challenge where it chose target species. These things aren't trivial. If you're going to have people look for fungi, you got to actually prepare information. So, we prepared these wonderful PDFs with information on what the thing looks like, where its projected range is, when you might find it, what you might find that's could look like it, to really try to help increase the chance of getting good data. And, not surprisingly, we expanded the record of a bunch of things. What was interesting, though, we also used that in the first iteration to see from a social science standpoint, what does that do? Does that help engage people? Does it make a difference? And so we asked the question, did the challenge inspire you to go out and look for those species? And 58% of them said, yeah, it made me go out and do a special search for those species. And then we asked, did the Rare 10 challenge, make you more aware of the need to document rare and threatened fungi. And again, over 50% said, yeah, I wasn't aware that there were rare fungi, that we had to do something about it. So from a social standpoint, we actually increased the awareness of people additionally, and we were excited about that. So these community science programs have expanded beyond pretty macrofungi, pretty mushrooms. This is a study that's being done in the Netherlands where they're asking people to get soil samples and send them into the lab, and they've been discovering all of these beautiful fusarium, not things that you normally think of people going out and collecting. There's at least 6 publications that has come out of this, a number of new species, new records, whatever else. There's now also projects where they're looking at having people send in leaves, and they're looking at leaf-associated fungi. So this whole bit of expanding how citizen scientists can be involved is really growing, and it's great. So that's kind of one major data source that we're really trying to take advantage of. How do we enhance engagement and enhance impact of community science programs? But there's another new data set that's out there, relatively new, which is environmental DNA. What we mean by environmental DNA is you go out there, you take a soil sample, or you take a bit of leaf, whatever, and you extract the DNA, sequence a bit of that DNA, and then try to match it to a library of those sequences. And so for these cases, you don't need the fruiting structures. You don't need the mushroom or whatever else to get to it. And so it has great possibility to do this. And it's starting to be used now for conservation. So there was a study where they were really great looking at kind of restoration work. Does the restoration work? So they go in after they did the work, can do the eDNA, see if the species, the targeted species, persisted and are there. There's a DNA study where they looked at documenting the diversity of fungi, in this case along Spanish cliffs. So has great potential. But there's also real challenges with using these data. And that's partially because of, well, 2 things. One is I just was at a talk that somebody gave, and most of the rare species aren't picked up, right? Not surprisingly, if you have a truly rare species, the chance that you're going to pick this up by taking, a couple of little soil cores isn't too high. I think the bigger problem is potential false positives, right? We all know that this DNA is very sensitive, and so you can have spores out there and pick it up there, or you can have an individual that's trying to get established, but it may not persist. Think of it from a plant perspective. You've got seeds being thrown around by birds all over the place that may expand its range, but it's not necessarily going to become established and persist. From a conservation status, we really want to know, okay, where are there mature, persisting populations? So as an example, which I find really interesting, and I don't know if somebody wants to talk about it with me, be great. We've got this species, Brigidoporus nobilissimus. It's one of the rarest species in the US. It is the largest. It can be, like 3 meters across in diameter, weighs 300 pounds. The bracket, there's only, I think it's 105 conchs known in, grows only on stumps of, 300, 400, 500-year-old fir trees in old growth forest in the Pacific Northwest, okay? Critically endangered. They went out with an eDNA study, and it's like in all the trees around there, it's all over the place, right? So they say, oh, it's not rare. And we were saying it's this rare fungus. But it only sporulates, it only completes its life cycle on 300, 400-year-old trees. we're not growing 400-year-old trees right now, right? So how do you deal with that data from a conservation status, right? From it exists in mycelial form or spores or something. It's being picked up in all of these trees, not just fir and other species, young saplings. So it's there. We don't know how it's getting around, but it's there. But to be successful and actually reproduce, it's going to take these old growth forests. So really interesting questions that come about with eDNA. Another story, it's not a fungal story, but it's one that means a lot to me coming from Chicago. We've got this problem with this carp that's in the Chicago River. Okay? Some of you may know that the Chicago River was reversed. Anyway, the Chicago River can flow into the Great Lakes. Currently, the carp is only in the river. It is not in the lake. And they don't want it in the lake because it's going to devastate the whole lake ecosystem because it's a ferocious, horrible fish. Evidently it tastes good, but from an ecological standpoint, it's a horrible fish. So they put up these electronic fences, barriers, at the mouth of the river. And then they keep sampling for DNA. And they keep finding DNA. But they've never found a fish, right? And so the argument is that it's either sperm or scales or something like that. And so the fish still hasn't broken the barrier, we think. Well, the same thing's happening with eDNA for fungi, right? You can find these eDNA, you can find these samples out there. Doesn't mean that you have a growing, reproducing, established species. So we still have a lot of conversation, working with some folks in Estonia right now, trying to think of some standardized kind of, what do you want to call it, guidelines for using eDNA for conservation work, because we don't really know what the best way to do it. We don't get all these false positive and some things that really are rare. All of a sudden, the eDNA says, nah, don't worry about those things, because we're finding it other places. Come on. What happened? All of a sudden it stopped on me. It likes these dead fish so much. It's frozen up. You don't want to see me give my pantomime talk the rest of the way. Imagine if you see, yeah.
Try again.
There you go.
I'm going to stay here for a little bit. Just
okay. So we're getting all these great new data, right? Either from citizen scientists or eDNA. That's great, but still the process of doing the assessment takes time. And so there's several initiatives now to kind of partially automate that work. And so the one that's kind of baked best so far is the rapid least concern that was developed by a team at Kew. Been used for plants, probably been done for ebony, I bet, right?
Not yet for ebony, but
okay.
It is something we are looking into.
Okay, but it's been developed for plants and it seems to work relatively well. Basically, it automates and can identify to some degree of certainty species that are probably this least concern. And therefore, the scientists then can focus on species that actually need conservation effort. But it has got some limitations. It's primarily looking at just distribution of the species. So we're partnering with Steve Bachman and others at Kew to see if we can modify it for fungi. We're not there yet, but we're just getting started on that. And our test case is going to be North American mushrooms, because we have, as I said, the mycoportal. So we have a list of all the species and the data from all of the collections in North America, and we can use that as our test case. So hopefully next year I can come back and tell you how well that worked. And then there's a newer tool called Redlist R which is adding to that. So it's not just looking at area, but it is putting in data on forest canopy change and other things that's really much more sophisticated. And so hopefully that will even speed up the process and the quality of the automated assessments even more. So I just brought that up just because that's where we're going to, is trying to speed up the process, because as we get more and more data, that's great. But then we have to do something with it. And so that's what's happening with that. Let me just switch gears quickly. Because I've been talking about red listing, and red listing is important, right? We got, as I said, it's useful for prioritizing action, but it's not the end. It's really the start, right? The idea of red listing really should be to start to raise awareness, enthusiasm, and information so that you can do something with that information. So we are starting to, for those of you in the Mobot plant group, these are going to seem a little bit less than what's going on in Madagascar or someplace, but it's a start. So I was contacted by this guy, Basil Gillot, and he's coordinating a women's group in this national park in Mozambique. And so trying to figure out, okay, how can they ensure that they're collecting these things sustainably? And so we're actually setting up a couple of experiments of, different size plots and how they're going to sample them and stuff like that. So really looking at those information to see, to ensure that these women who are now using these not just for a supplemental food source, but more importantly, as a supplemental income for their families, can continue doing this into the future. This beautiful fungus, Hypercreopsis amplectans, are the tea tree finger fungus, is this common name, has been known only from a couple of twigs in Australia, mainly in Victoria. And very rare. After we were able to assess it and give it a nice critical endangered, they could use that to obtain funding and use that to do work. And so here's from Fungal Map, but there's other things I could have showed where they're now working, doing much more sampling. They found another population on one of the islands off of Victoria. They're trying to get it in culture to do ex situ conservation. And trying to understand its ecology better so they can reintroduce it, okay? All that happened because we had the red list data to document that species was in need of attention. And the last example, I really like this example. This is Lojo, Butyroboletus lojo. It's an endemic bolate from Chile. It forms mycorrhizas, or it forms this mutualistic relationship with Nothofagus. And it's a tasty edible, and one of its major threats is unsustainable harvest. And so it's declining because people love it and they're collecting it a lot. And so after it was listed, they were able to pull together, and those of you that are in the conservation field know how challenging this could be. They pulled together a collaboration between the National Forestry Institute, one of the main universities, an NGO, and the local people, the Mapuche Indians, to develop a whole integrated program. So changing how they're sampling, but also looking at where these are. You can see the women up there measuring the trees to understand its ecology. They're trying to get in to do augmentation, where they take some of the mushrooms and actually are grinding it up and putting it back into the field to increase the number of individuals out there. And what really I love about it is not only it seems to be successful, but the engagement of the local community. So here's a Mapuche elder holding a lojo, and it really is a cross-generational project. So it's been really exciting to see that develop. We're also working to raise awareness of fungi and the need to include them in conservation work. And some of you may have seen this. We're calling it the ThreeF Initiative, fauna, flora, and fungi, to get that on board. And so we do have at least one of the largest conservation groups to buy into this. So Rewild, the NGO Rewild is doing it, and IUCN now is using the 3Fs in all of their work. So remember, if you're talking about biodiversity, no more of this plant and animal stuff. It's plant, animals, and fungi, or it's fauna, flora, and fungi. You know, so fungi get up there, right there on par with plants and animals. And then from North America, I don't know if anybody's on from NatureServe on this call, but we're working with NatureServe, state heritage programs, whatever else, to figure out how we can provide help so that they can include fungi in their management plans, add to their rare and threatened lists. And lastly, also trying to drive the fungal ecology community to really start looking at some of the issues that are involved with fungal conservation. So really, this was a paper we did a couple of years ago. Tom May took the lead in conservation biology, where we really kind of identified some needs. One is, conservation biology, our actual discipline, focused discipline, would help facilitate framing research questions. Fungi, we recognize that many groups of plants and animal folks don't recognize it, but no organism lives on its own, right? They're all living in partnerships. And so what we're saying is that we may need to be shifting our conservation efforts away from single species things to look at relationships, right? And so really figuring out how we can move that and how we can look at, okay, if this is a species that is associated with this tree or whatever else, how do we do a management plan that benefits both? So that type of thing. Then also recognizing, there's always been saying that, just save the forest and we're okay. But we know that there's certain forest management practices that can be tweaked that will benefit the fungi, not hurt the forest. So what are those forest management practices that are needed to ensure the long-term fungi? And some of them have been put into place, and some of them were geographically. So until recently, Europe had a tendency to pick up all of its dead wood. It actually manicured their forests. So they were losing out all of their wood decay fungi, all of this kind of stuff. US has been messier all along, so that hadn't been as much of a problem in US forests, but it still was a bit. But we recognize the importance of coarse woody debris. The other thing that is now recognized is the need, when one is clear cutting, to leave basically mother trees standing around there, because they're the source that maintains the mycorrhizal fungi to do this. So really some different methods to change how forestry practices are being done to ensure that fungi are being taken care of. And then really just more fostering that conversation between conservation mycologists, researchers, and practitioners. So in conclusion, work needs to be done, a lot of work needs to be done for documenting fungal diversity, distribution patterns, conservation status. And much of this data is observational, what occurs where and when, how these patterns are changing. So, coordinating citizen science programs to enhance the engagement and the impact's important. Figuring out how we're going to use eDNA data in there appropriately to give us those extra data are really things that are going to be changing our ability. And then hopefully developing some automated tools to speed up the redlisting process. But again, while redlisting is important, we need to put more into the recognition created and the data generated to put that to work. So just to end, my new soapbox is that we need to improve the importance of fungi. It's high, but it needs to be higher. Right? When I talk to conservation groups, government ministers, we're to the point now that they recognize the importance of fungi. They said, oh yeah, fungi are important, I get it. But we don't have enough time, money, people, or whatever to do it. So we can't afford to do it. And I'm working to change that to they recognize they can't afford not to do it because of the importance of fungi. So with that, let me end on some beautiful North American rare fungi, and I'll take any questions you have. Thank you.